I have, something must have happened. Yeah. But not everyone feels comfortable telling what the hell happened, but.
So like, the bio but my problem, no one like, you didn't.
Sometimes, not always, we don't talk to each other necessarily.
But you do know what happened.
I got no connection, bro. I don't know why.
Like, I was very forthcoming when
We went to that little
Yeah.
session that she had, and
that.
most of the people that were there had the wrong data.
Yeah.
Yeah.
I was appreciate, I did
Data for one.
some way.
I looked at it and I'm like, this is wrong. Look at the, so she uploaded the right one. And then the people that had left.
And so the following week, we're like, we are doing it right now, doing a lot of things similar to that. But I was gonna get a PET scan, oncologist, this and that, so we went remote
One.
and switched to a
Stay up
case
till like 6 in the morning.
Till
Dang.
started, so. And then when it started chemo.
I know you told me, but you responded, I'm like, what?
the
Yeah,
day
I had to be, I was like,
after
this
chemo,
is terrible.
So my Friday section went remote, the others kept in person. But, yeah, but I struggled that semester, and the following semester, my goal was to stay in person. And I ended up, or I wanted to go to get to at least from here, and I couldn't go. So like a couple of weeks before spring break, I wrote him, like, I'm sorry, I wanted to get this as much in-person experience as we could, but I just didn't.
I think.
I was in and out of
the thing
hospital
that
so
she's
much.
looking for, if you
There
don't
was
have to
a lot.
interpret your data.
And
Like,
for
she's not
me,
looking at the
you guys see that I'm the one prepping for most of them, and then sometimes for the others too. I couldn't physically, I was using a walker.
That's what I'm saying.
That's not
You
so
did
much.
that, right? You should
I
be.
had to switch from.
So she grades it leniently.
Wait, and
I don't know about
Yeah.
leniently. I feel like she relies more on the peer, to be honest with you. It's more on the peer. I was like, ah, she fucking really like big circle. Scrolls down, next.
Thank you.
Oh my
oh no.
No.
I only made it to class.
I did it
It's called the-
yesterday morning. I was awake and I got up to go and I almost fell.
Oh, no.
And I was about to pick it up and I'm like, yeah, if I try to drive. I think, I don't know what it was. I just was like really, really sick. And then the day before I threw up on my way to school. So I'm like,
Yeah.
yeah, let me not try to.
It was, yes, like multiple times. of going around.
I have the dispose of my computer.
Exactly, so I should actually. Go. Intensive, I feel like at this point, but I had this on Friday, I was like so exhausted and I had very busy stuff in the lab, I was doing several hours. And eventually I just couldn't take it and I went home. And I was supposed to go pick up my daughter, and when I got up from the bed to go get her, I just collapsed. Like my husband had to take me to ER, collapsed.
Really?
Yeah.
Were you in the ER?
Yeah, and then I ended up staying overnight.
What did they say? What was wrong?
So my body is still recovering from chemo, so it has other issues along with it, right? But maybe I was getting a bug and it affected my lungs. My lungs aren't very good right now anyway, that it caused super, my pulse oxygen not good, like, you know.
Did you hit your head? Did you like hurt yourself or did you just fall back on the bed or something? Like, did you go on the ground?
I fell on the ground, but it was carpeted, so carpet, so it was fine, very nice and soft.
You're just like, I'll stay here. It's comfortable.
There and like we've been taking care of her the last couple of weeks and she can't get up physically. It's impossible. And she just like started screaming. I hear her. I want to get up and say I'm okay, but I couldn't.
Oh my God.
But yeah, but yesterday after our lab, when I was in the lab yesterday, I had the same thing. I was like, I had really pain and body aches and just, I was having a really hard time. I was just struggling. And so I ended up going home early instead of finishing the prep because I was like, it's not gonna happen.
Yeah.
But yeah, but I also seem to be coming down, actually, clearly, I have something, congestion in my. Let's see how it goes there. Guys. Well, so remember we started talking about signaling, my favorite, well, I have a lot of favorites. But how we communicate inside the cell and how we talk to each other in the cellular world. And we started just kind of briefly reviewing what different ways molecules can attach to cells and elicit responses. And we're gonna go through a lot of that today. And this is, I believe this is where we stopped last time. Correct me if I'm wrong. That we were talking about how sometimes you'll have a molecule that will bind, some signaling molecule, it will bind to the cell surface receptor, never go in, and it's gonna elicit the response through this binding interaction because this receptor is going to send that signal along with the change in conformation or anything else. On the other hand, other times you have molecules that do permeate through the plasma membrane and they go into the cell before they bind to their receptors and then elicit a response through that mechanism. So in this case, you have the intracellular receptor. A lot of times, when we think of receptors, we think of them hanging off of the membrane. They don't have to be that way. They can be intracellular as well. They can be in the cytoplasm. And then once they bind to their signaling molecules, they will usually change conformation and either go into the nucleus or somewhere else to have a response. Yes, dear.
I was just gonna ask if you can dim the lights because it's
Dim the lights, yes, I
kind
can
of hard
try.
to see it.
They were like dim only the front ones.
Yeah, I wish, no, it's not an option, unfortunately. That is something that we are trying to get them to do though.
It does make more sense.
I know, but. Oh,
Did
did it?
Did it?
It did it. Is that good or do you want more off
I think
More.
that's good. I just can't, I couldn't see before.
More.
But it's
at all, right? I know you can see without it. Or. Anyone else?
What's this?
Yeah, because that's like.
Maybe don't pass out.
Ambient environment for sleep. I don't like it either.
It's not my problem. It's last time.
Sit me up front, so if you want further
Was it
down.
some class the other day?
Or back. Sure. Whatever you guys want. Oh, my God.
Oh, yeah, good night, bro.
Very good.
Oh,
Not a good choice, obviously. Do it again. Like, come on, do something.
oh, there you go.
Is that good?
Yeah.
Okay. Sorry about that.
It's like American.
That's great. Thank you.
So sorry.
See.
Okay, so, next part of that is, well, depending on where the signal molecule is gonna bind and how many mediators it has to pass through before you get the final response. The response can obviously be fast, where it's immediate to, within minutes, or it can be slow, where it may take even hours to get to the actual response from that cell. And you can think about what those responses might be, that might be slow or fast, right? So if all it needs to do is, once it binds, it needs to make the protein activated, right? Just a quick phosphorylation and now the protein is active and it can do work. That would be something that can be done very quickly, and that would be considered a fast response. However, if whatever the signal is, the signal that is binding, if that has to now change the entire gene expression profile, maybe to induce cell growth or replication or differentiation, then that's gonna take time, right? That can't just happen quickly. It would need to send that signal into the nucleus so the transcription can change and new genes can be transcribed and then they will have to be translated. So that's gonna take some time before that can happen. So that's an example, obviously, of what that would look like. Again, it doesn't mean that a signal only can be fast or slow. You can have some signals that can work both ways. They have a quick response initially, and then they have a long-term response that happens over time. Okay. So how do these happen? Well, essentially they happen because the original signal is going to affect many mediator proteins as a result of their activity or their binding. So these extracellular signals will, let's say, bind to a receptor protein. That receptor protein will then have either activate or deactivate a mediator protein. And those will then kind of play a relay of sorts, right? So each mediator protein will affect the next protein in the pathway and the following protein, eventually leading to one of these three key target responses. It can alter a metabolic pathway to either activate it or deactivate it, right? It can alter the shape or movement of the cell depending on what it needs to do in that environment, in which case it's going to be directly affecting the cytoskeletal structure and proteins related to those. Or it could be a transcriptional regulator where it's going to alter gene expression and lead to further changes downstream for that cell's growth, replication, or differentiation. Those are typically the three things that will happen. This initial signal that you receive is called the primary transduction. That is, the signal binding to a receptor protein, wherever that may be, intracellularly or extracellularly. And then in those mediator proteins, all these proteins that are talking to each other in response to it, this is the process of transduction and amplification, because each time this one signaling molecule binding to that receptor will lead to activation of multiple mediator proteins. Each mediator protein will then activate multiple second mediators and so on and so forth. So you're going to end up with large amplification of that signal just by that single molecule binding to the receptor. So it's not a one-on-one response. And so this is a way to amplify your signal as well. And then your feedback is your final response that is controlled by your effector proteins. Okay. So we really want to be able to identify and understand these three main groups of signaling or signal transduction pathway. The primary transduction, where the original signal is binding to its receptor, the amplification of signal through mediators during the intermediate steps, and then the final feedback, which is through the effector protein leading to the response. Yes.
So is that receptor protein like sending like this intracellular signaling molecules that then go?
Yes. So this extracellular signaling molecule in this case is shown binding to a receptor outside. It doesn't have to be that way. It could have been that it went in and bound to a receptor inside, but that initial binding of that signaling molecule to a receptor, extracellularly or intracellularly, that's your primary transduction, right? That's your step one. Okay, so this response, like I said, is going to be conducted by your effector proteins, and those are going to be in the shape of either altered metabolism, expression of genes, or shape and movement changes. Now, these are going to be specific as, again, not just controlled by what signal is binding, but also what cells are in that environment. If you have just one type of cell culture, it doesn't mean that all of them are going to end up responding the same way. So there are sometimes you have these contact-dependent signaling controls where cells that are not all bound to each other will be affected one way, and cells that have contact with other cells all across will be affected a different way. And here is an example of that. In this case, you have these epithelial cells. These are all unspecified epithelial cells. So these are not totipotent cells. They are kind of a subset of pluripotent cells. Remember last time we talked about how you can have the cell population in each organ system that is specific to that organ alone, right? These are like tiny population of stem cells. They're partially specialized, so they can still be made into multiple cell types, but within that organ system. So this is an example of one of those. So this is those, partially specialized epithelial cells. And these are going to be they can be made into a nerve cell, or they can be made into the supportive structure, either one of them. So in this case, when you have cell specialization through receptors binding to each other, what you will get is that the initial, in this case, there is a signal that is a inhibitory signal, and it is membrane bound. It is called delta signal. And if it comes into contact with its receptor protein, which is the notch, it's the receptor portion that is going to bind to this delta signal. And if it is bound on all sides, all the delta signals are bound, you're going to end up developing that cell into a nerve cell. All these surrounding cells remain in the supportive structure. They remain this unspecialized epithelial cells. Only the cell in the middle where every single receptor was bound or every single delta molecule was bound is going to be turned into a nerve cell. Okay? But all these other cells, they are not going to be able to do this. So this is a way that we can specialize some population of our stem cells into a particular function, into a particular type of cell, while the others remain the way they were as initially. Does that make sense? In this case, by the way, so we did talk about different ways that you can have these signaling molecules. What type of interaction is this? So the delta protein, the inhibitory signal, is also embedded within the membrane, and the receptor is embedded within the membrane as well. So I will show you. Yeah. What type of signaling is that? Exactly. It's a contact-dependent signaling, and it was written over there too, but I want to remind you that in a contact-dependent signaling, both the signal and the receptor are bound to membranes of adjacent cells typically, and that's how they are interacting with each other. It could sometimes be the signal is bound to the extracellular matrix through some interaction and then interacting with the cells, or it would be another cell with the receptor and one cell with the actual membrane bound signal. Okay, so there are several small hormones and small molecules that can also act as intracellular signaling molecules. Actually, most of the hormones behave as intracellular signaling molecules at the end of the day, whether to control growth, metabolism, reproduction, any of those cases. And a lot of them for metabolism as well. So cortisol, estradiol, testosterone, thyroxine. Again, this would be part of your stress response, for example, metabolism, and then sex hormones. They are all essentially transcriptional regulators, and they are going to directly affect intracellular signaling and then gene expression downstream to relate to changes in that particular cell's behaviors. So again, you can have those extracellular signal molecules binding to a receptor. You can also have intracellular signaling molecules that can bind to receptors as well. And this is just, again, reminding you of that particular structure. This shows you how you can have that amplification effect. So you will have these initial relay that is going to help to transmit that signal. And then at some point, one of the mediators is going to then either secrete further signaling molecules, right? So this is the second messenger. This was, in this case, the first messenger. This is the second messenger. And the second messenger is released in a large enough quantity that now it can amplify that signal a lot by several fold and affect multiple cells in response with it, or multiple parts of the cells in response to it. So this would be an example of amplification. Now, once you get the final response, well, at that point, you have two choices. You can either continue the cycle and get more of the signal moving, or you may not want to do that, right? You may want to shut down the system at some point and not continue to have the cycle keep going. Because remember, if the signaling molecule is still attached to it, it could technically continue this process and continue to amplify the signal again and again and again. We may not want that. So you many times at the level of your final protein that are effector proteins, you have a molecule or those final proteins, the effector proteins that are made, they will act as feedback for the whole pathway itself. It can be a positive feedback loop or a negative feedback loop. In the case of a positive feedback loop, these proteins will further activate those second messengers and continue this process along, continue to make this process go forward. In other cases, this process will be inhibited by a negative feedback loop, where once the effector proteins are made, they will directly bind or directly affect those second messengers and shut them down so that you cannot get more activation of that system. And you just get that first pathway and that's it. Yes.
So for these intracellular signaling proteins, does there need to be an extracellular signal first that sets off this?
Say it again.
So in this like image, it shows that there needs like an extra site.
Yeah, so in this case, it's an extracellular signaling molecule. So again, part of what cell signaling is the how we respond to environmental stimuli or stimuli for things around us. So that's what it is indicating. Again, that extracellular signal doesn't have to remain extracellular.
Yeah.
It may be binding to the receptor outside or maybe coming in and responding to that inside. This is not an intracellular messenger molecule, right? So this particular pathway, this is only looking at signals coming from outside and received by the cell extracellularly or intracellular. We'll talk about things that get affected on its own inside the cell later, okay? Yes.
So you said that the small messenger molecules are the
Yes.
feedback regulators?
So no, these are the small intracellular messengers. These are second messengers, right? So this is where your first messenger. This is your second messenger. This is used to amplify the signal. The feedback loop that happens usually happens by the effector proteins. So, once you go far enough into the pathway that you already created the first effector proteins and they're going to directly affect gene expression, they will then themselves regulate whether or not there needs to be more created. And it will depend on the pathway, whether they are going to be controlling it positively or negatively. And we'll see examples of both of them, right? So again, this talks about that positive versus negative feedback. In a positive feedback, the final, the effector proteins that you are getting, they are going to be relaying that message back to that intermediate process, right? Usually the second messengers, to continue to amplify that signal and to continue to send it forward. In a negative feedback, once this effector protein is made, it's going to negatively regulate that second messenger and it's going to inhibit it from going further. So it's no longer going to amplify that signal and it's no longer going to continue to process it. Okay. So by doing that, these intracellular signaling pathways or proteins can essentially act as molecular on-off switches, right? So it's going to tell you how long you want to keep that activation going and when you want to switch it off or on. And they can do that by two different main ways. One is through protein phosphorylation, right? And we already talked about phosphorylation being important for activation of protein activity or deactivations. And then also through GTP binding proteins. So these are two main mechanisms through which intracellular signaling proteins can act as molecular switches. In both cases, whether it is through the phosphorylation, through ATP-dependent protein kinase activity, or through GTP binding proteins, it's the same idea, essentially, right? So in this case, your original protein is switched off until you get a signal which leads to an activation of a protein kinase, which is the intermediate signal, that is going to take ATP and convert it to ADP as it phosphorylates this protein and activates it. And that is then going to be the active protein working the system. Once it transfers that signal further, it is going to become dephosphorylated from protein phosphatase activity and go back to its off position. And this will continue as long as there is signal coming in to activate the protein kinase and cause this activity to happen. Yeah. In the other case, you have a signal that is through GTP binding proteins. In this case, in the off position, the protein already has a GDP bound to it, okay? So the GDP, when the signal comes in, is going to change its conformation in a way that now it is going to release the GDP and takes on a GTP, which is going to have that extra phosphate because it's a triphosphate instead of a diphosphate. And that's going to activate the system. And then through hydrolysis of the GDP, you will release the phosphate and go back to your off position. And this hydrolysis happens as it transfers that signal to the next protein in line in the relay. Yeah. Yeah, goal yesterday to make up for it today. So, this GDP, the ATP hydrolysis, you guys have seen multiple times, and it's a little bit easier for people to go through. This one is something new, and so we're going to talk about it a little bit more in detail. And this one actually needs other proteins bound to it and working together. This process is not by itself. So the individual, GTP hydrolysis as well as this transfer from your GDP into GTP takes helper proteins. This is regulated by other proteins that are interacting with it. So you have two different proteins that you need to know. One is the GAP protein, which is GTPase activating protein, just like it says. And then the second one is GEF, or GEF, which is guanine nucleotide exchange factor. And that's the one that's going to exchange the GDP into a GTP when it needs to activate it. So you start off with your inactive, protein. In this case, this is an inactive monomeric GTPase. And so it has a GDP bound to it. When it is, when it interacts with an activated GEF, this guanine nucleotide exchange factor, this exchange factor will kind of knock off the GDP and put a GTP in its place. That's basically the entire job of a GEF, right? This nucleotide exchange factor, all it does is it exchanges the GDP to a GTP. And by doing so, it activates the protein. Yes.
So the phosphate doesn't come and bind to the GDP, it's exchanged for.
It's exchanged for an already active GTP, right? So it knocks off the GDP and it puts in a GTP in its place. So it's different than the ATP hydrolysis where it knocked off the phosphate, right? To get to, that was a good example, right? So in here, it converts from ATP to ADP as the phosphate is moved over. This is not the case here. Here, the entire nucleotide along with its phosphate is moved over. So you knock off the GDP and you put in a GTP. That's its job. And that now becomes activated. Once it's activated, then a GAP protein, the activating proteins, is going to activate its GTPase activity, which is again similar to this protein phosphatase activity, same idea. But in this case, it's going to activate it so that this phosphate comes off and now you're left behind with the GTP. And this phosphate usually will then get transferred. It doesn't just come off, it goes to the next protein in line. Okay, that it is going to control. Yes. Single unit.
Okay.
Wait, what did you say? It goes to a different, what goes to a different molecule?
This phosphate, like this phosphate is not just going to be released, it's going to go to whatever the next mediator protein is in the process. It's not just floating around in the cytoplasm, it's actually going to be activating the downstream proteins in most cases. So that type of effect is present in many intracellular signaling pathways. And we will, as we talk about more pathways, you're going to see them getting affected by these. There's a whole class of proteins called G protein receptors that we'll talk about later, I think today, otherwise next time. But those G protein receptors, that's how they work. completely through these G protein activities. So if you look at the cell surface receptors, they fall into three main classes. One of them is these proteins that are going to work through the G protein, GDP, GTP activity. They are called G protein coupled receptors. The second one is enzyme coupled receptors, where you have an enzyme that is going to stimulate or get activated when a signaling molecule binds to these receptors and become activated because of them. And then the third are your ion channel coupled receptors, which we've already talked about a little bit, right? But we'll talk about them more in detail. And they allow ions to enter into the cell or exit the cell based on what the membrane potential is and what the function is that needs to be performed, especially during like action potentials when the electrical signal needs to be moved on. What happened? We're going to talk about them again. So it's ion channel coupled, G protein coupled, and enzyme coupled. So let's talk about some extracellular ligands. So these are your signaling molecules, as extracellular signaling molecules that you can be binding to in order to activate any of these effects. Now, you don't need to know any of these as such. These are just given to you as an example, but given information about a particular molecule's effect or a particular molecule's action, you should be able to predict what type of molecule it might be or which one it might be, okay? So yeah, so you have these different ways that they can be activated, right? So the substances, like if we have barbiturates that we are taking, or we are taking those sleeping pills or anti-anxiety pills, right? Those are usually going to activate the GABA signaling pathways. GABA is a neurotransmitter, so it can directly affect ion channels because it's going to propagate those action potentials. And in the end, the effect is going to be relief of anxiety or increased sedation. Similarly, nicotine is going to bind and produce this acetylcholine as its signal. This acetylcholine is going to activate ion channel coupled receptors as well. And we talked about how one of the functions they can perform is vasodilation or vasoconstriction. And that's kind of what it's going to do. And it's going to lead to changes in the blood pressure, right? So again, these are the most common ones that you will see. Morphine and heroin, they are one of the big examples of G protein coupled opiate receptors that will go through that G protein pathway to give you pain relief, right? And others like that. Now, look here, the curare and nicotine both release acetylcholine as their signaling molecule, and both of them will affect the ion channel, right, coupled receptors. But because of the receptor that they activate, that receptor is different, so the effect is different. So in curare, you have constriction of the blood vessels, or in the neuromuscular pathway resulting in paralysis. So curare, do you know what that is, by the way, curare? It's a poison. And then the nicotine is going to constrict blood vessels to give you higher blood pressure. So this is affecting the muscles and their signaling molecules through that, directly affecting the their ability to constrict and relax, as well as the nerve endings that are coming. So yes, so this is through with that. Here are other examples of signaling molecules that are intracellular. These are ones that may be produced by our bodies themselves, by the cells themselves, either right there or in nearby environment or somewhere in the body of that organism itself. So these are your hormones, and you can see their site of origin will be in different places. Some are going to be right at the source. Some are going to activate things away from the source. And you can look at how that might affect this is the final target response that you will get from them. And this again is another set of molecules that are also present inside the body. Most of these are present inside our body and are going to be produced by the organism themselves. They could work to help to grow particular types of cells. They could be involved in inflammation response or muscle relaxation and constriction. They could act as neurotransmitters like GABA and acetylcholine. Or they could be contact-dependent signaling molecules like delta, which are directly involved in differentiation of cells. Okay, so growth, reproduction, neurotransmitters, and then differentiation. So let's start by talking about G protein coupled receptors, since that's what we were just beginning to start with. G protein coupled receptors are embedded within the plasma membrane, and they are going to be typically multi-pass domains inside the membrane. Typically, there are 7 transmembrane domains in a typical G protein coupled receptor. They have an extracellular and an intracellular portion that is going to directly interact with either the extracellular environment or send molecular signaling down the pathway. Here is an example. They'll have a ligand binding site in their extracellular space where signaling molecules will bind. So how do they work? They will work in the normal state. You are going to have your inactive receptor proteins embedded within the membrane close to an inactive G protein that is also present within the membrane. Now, G proteins are anchored to the plasma membrane. They are not integrated within it, okay? So that's different than your receptor. The G protein receptor is completely embedded within the plasma membrane. On the other hand, your G protein receptors or G protein itself, the inactive G proteins are going to be just integrated within the plasma membrane and not embedded within them. Now, they come as 3-unit system. You have an alpha, beta, and gamma. The alpha unit is where the GDP is present, okay? So that's the main unit that is going to have that transfer of GDP to GTP activity. And then the beta and the gamma are the ones that are going to help with that process along. So when a signaling molecule binds to the inactive receptor, the G protein receptor, it's going to cause that activated receptor to interact with the alpha unit of the inactive G protein. This binding will release the GDP and cause a GTP to come in its place and lead to an activated alpha subunit, okay? This activated alpha subunit no longer can bind to the beta and gamma units together, and instead will cause them to be activated and leave the main system. And now this can go and affect further signaling pathway downstream. So G proteins, these are trimeric proteins, alpha, beta, and gamma. Alpha is that GDP to GTP transfer protein molecule that is going to be directly activated by the G protein receptor protein, right? And this activated receptor will bind to it, lead to GDP dissociation, cause GDP to come in its place, activated, which will lead to dissociation of beta and gamma together as an activated complex. So then what happens next? We'll talk about that next. Here is an example of how a GTPase activity of the alpha subunit can turn off the G protein. So you have your target protein that it's going to affect. You have the activated alpha subunit. So we are starting here, right? You have the activated GTP bound alpha subunit. You have an activated beta gamma complex, 2 separate things, right? So now what is it gonna do? This particular GTP activated alpha unit is going to bind to its target protein if present within that system. This target is going to then lead to hydrolysis of the GTP back to the inactive GTP state, and it's going to, in its process, activate this target protein to do what it needs to do. This molecule here is still dissociated and activated. Once this GDP is there and it's inactive alpha subunit, it can bind back to the beta and gamma subunit to make that trimeric unit again. Have a question?
No, people always think I'm like staring at them for no reason.
Yeah, I thought you had like a question or something,
I
so.
just blankly stare. Sorry.
Were you listening?
I was listening.
Okay, that's good. I'm all good then.
I get that a lot, honestly.
Sorry about that. No, but it looked like he had genuinely looked like he had a question. Any other?
I appreciate what you're
question?
saying.
So this process continues.
This will continue as long as there is signal available. This is a one-time deal. It has to get reactivated by another signaling molecule. So it's not something that will continue on and on and on. Once this target protein has interacted with the GTP and hydrolyzed it and gone its merry way, it's inactive. That whole trimeric unit is inactive, back to its resting state. Everything is back to normal. To activate it again, you'll need another signaling molecule bound on top. You'll have to start this whole process again. So you'll need more molecules present in the system that can go in and bind again and activate again and go on. Okay. So some G proteins will work on target proteins that are within the plasma membranes. Other are going to activate proteins that will go intracellularly and may even transfer into the nucleus to affect gene transcription. So when they activate something on the outside, so here you have that target protein again, in this case, right? So you had a target protein here. So if you have an activated alpha subunit, when it binds to its activated protein, or its target protein, this target protein now gets activated as well. In this case, it's an enzyme. And so it's going to do what it needs to do. It's going to, work on whatever molecules it works on. And so it will take its substrate, it will break it down. And typically these breakdowns are the second messenger molecules that are going to be that amplified signal that's going to diffuse through the cytosol and bind to other proteins and affect them. So this is how it's transferring, because remember, this G protein complex is never leaving the plasma membrane itself, right? It's stuck there. So whatever it needs to do, it needs to send out that signal through other messengers one way or another, whatever they may be. So here, the activated enzyme is membrane bound. The actual activated G protein is membrane bound, but they can now send those signals inward to the rest of the cell through that activity of that enzyme, right? Other G proteins can actually regulate ion channels. So in this case, you have acetylcholine that is acting as your binding partner for your G protein receptor. So it binds to that, it activates it. It's going to go activate the GTP, right? By putting the GTP, so it's going to activate the alpha subunit, and it's going to activate the beta gamma complex. So this beta gamma complex is going to, in turn, bind to the potassium channels and activate them. So each part of this G protein, complex can have its own activity. So the same G protein activation can lead to one response through the alpha subunit and a different response through the beta gamma subunit. Cool. Another way that it can do that, again, so this was one of those, again, environments where you have that plasma membrane. This is the intracellular portion, that's the extracellular portion. Here, again, another example of that. Now you have this beta gamma complex that is bound to the potassium channel, causing it to get activated and open up. Again, these are acetylcholine molecules that are bound to it in this case. And you have your GTP activated alpha subunit. Well, this is going to get inactivated when that membrane potential is maintained. It reaches that equilibrium where the potassium channels shut down. When they shut down, you will get your inactive G protein release, because that is going to be inactivated by the hydrolysis of this GTP back to GTP through the GTPase activity. And then you'll get your trimeric G protein in its inactive state and a closed potassium channel as a result. So this is another way that you can regulate your ion channels. Yes.
This might be a dumb question, but I don't remember if you told us what G proteins are, like what they mean, or what G protein means. What is a G protein?
A G protein is a protein that is regulated, that is switched on and off through the GTP to GDP transferase activity, not through the ATP activity. That's why they're called G protein. Yes.
No, that's fine. just kind of confused. So it's like, you heard C on the last side.
Yeah,
So I see that acetylcholine goes in to the G protein.
well, so that acetylcholine is going into that G protein receptor, right? Those transmembrane, the 7-pass transmembrane domain.
Yeah.
And it's gonna activate it, so it's gonna bind to this and activate it into this state, right? Where there is a GTP, just like before. So anytime, where, here. So consider this acetylcholine, it's coming in, it's binding to this complex, it is activating it by making it GTP.
Yeah.
And then you have the activated beta gamma complex that's gonna interact with the potassium channels, open it up. This one is going to get inactivated, and when it releases those phosphate groups, it's going to cause these channels to close and come back together as a inactive complex somewhere.
Yeah, it just kind of confused me a little bit.
Right.
I see it's so. this
Yeah,
is
they
just-
never show that binding right on top of each other, that's the.
This is just controlling the potassium.
In this case, it's one way to control the potassium. Yes, not the only way, right? We've already talked about other ways.
So acetylcholine also goes into the potassium.
It can, there are some where you will have that type of activity, yes, where you can have acetylcholine, and we've actually looked at some of those in another slide last time. Where we looked at how the same molecule can have different effect. Last time we talked about that. Should not do. It was back here. Different cells, different effects.
So how does it get from that receptor protein?
It doesn't, it's
Okay.
separate. There's
Separate.
more acetylcholine in the
Okay,
system. It's having a different effect, yes. It doesn't have to be just on one side or the other, and I'm not going through that. How are you? Right? No, sorry about that. Okay. Oh, you guys can take your papers. It's okay. Today we have hard topics. We have to talk. No stickers for
Huh?
You should just give us a sticker for being here.
Existing.
For existing.
Yeah.
I should get 2 stickers then.
I'll give you stickers.
Yeah, I'll give you, I just think it.
No.
We don't have the.
Yes.
Yeah.
Okay. Let's see, what are those second messengers that the G proteins create? Remember we talked about back here that sometimes they have to send the signal, and they send it through second messengers that they are creating through activity with activated enzymes. So there are some common ones that happen. One or 2 of them rather are using this inositol triphosphates and diacylglycerols as second messengers. So these are 2 important second messengers of G proteins. And they are the ones that are going to get broken down or activated and then sent into the body, into the, not body, but into the cytoplasm of that cell and activate other proteins. And here you can see them, both of them are membrane integrated as well. Remember, right? They were within the membrane to begin with. You had those phospholipase C and you had the inositol bound triglyceride chains, and then you had the diacylglycerols. Well, when you have these second messengers, they're going to get cleaved off, and a portion of them is going to end up into the cytoplasm and act as second messengers. So here is an example of how that might happen. This is using the inositol phospholipid pathway. This is how you get a rapid increase in calcium ions in the system that can then act as its own activation of further pathway. So you have your signaling molecule, it's binding to the GPCR, so the G protein coupled receptor. It's going to activate the GTP, right, trimeric complex. So you have the alpha and then the beta gamma subunit. The beta gamma subunit is going to bind to a phospholipase C and activate it. This activation does exactly what it says that it will do. It's going to break down, right? It's phospholipase. So it's going to break down these inositol phospholipids and release the IP3 portion from those lipids. So these inositol triphosphates are going to be running around And they can bind directly to calcium channels and cause them to open and release calcium into the cytoplasm from the ER lumen. Okay. Another way that they can also have further activation of the system is by that leftover diacylglycerol. So that DAG molecule that's left behind after the inositol triphosphate is cleaved off can bind to activated PKC. This calcium, when it's released, binds to the PKC. These are kinases that are going to then, once activated, bind to the diacylglycerol, and then they can actually finish this loop and put this back on and life will go on. Cool. So this is one of the things that happens during fertilization of egg. When that sperm enters the cell, at that moment, you have sudden rise in cytosolic calcium in the egg. This is through exactly this process, through this G protein coupled receptor activating the phospholipase C and the inositol triphosphate. And as it does that, that kind of shuts down any other entry of further sperm from outside and shields it and makes it the final cycle that you see. There are videos that you can find actually online, and I might have it linked somewhere in there as well, from fertility clinics where they examine the health of the, the embryos that are forming. And one of the first indicator of their health is how strong of a response they see with the calcium ion signaling. It shows up as like this bright spark at that moment of fertilization. And what they have noticed is that the brighter the spark, the healthier the embryo will be from that particular union. So they can look at and kind of even, guesstimate which ones are going to be the healthiest embryos without any issues, and that will grow more normally. Okay. Other responses that are mediated by phospholipase C, so the same kind of idea, right? Same enzyme that is also breaking down the inositol is also in the other systems within ours. In the liver, you have the vasopressin, which is a peptide hormone that breaks down glycogen at the end of the day, and this is also controlled by that phospholipase C activity. Acetylcholine in pancreas and skeletal muscle will lead to secretion of amylase or contraction of the muscles, and that's controlled by phospholipase C activity as well. And then finally, thrombin in blood platelets will lead to aggregation of these blood platelets. So, cool. Now, another common second messenger of G protein coupled receptor is back to cyclic AMP. Is cyclic AMP that can also act as a second messenger in the system. So in this case, it's going to be coupling with an ATP AMP system, right? So remember there were two on-off switches. You have the G protein and the ATP coupled. So here you're gonna be coupling those two systems together. So your adenyl cyclases are activated by that alpha subunit, activated alpha subunit of the GTP complex. When it is activated, it is going to remove a pyrophosphate or two phosphates from the ATP and give you the cyclic AMP structure. The cyclic AMP will then act as second messenger, and it can go into the cytosol and even into the nucleus and activate gene expression or affect other effector proteins. So this is another important part of how G protein coupled receptors function in intracellular signaling. So in this case, protein kinase A is the main intracellular target for the cyclic AMP. Any protein kinase, when you're looking at it, their main job is to phosphorylate, right? That's what they do. They're kinase. The kinase activity is adding phosphorylation to something. So by presence of cyclic AMP in large amount, it signals, it activates these protein kinase molecules, specifically protein kinase A, and it can cause it to become active and then phosphorylate ATP, AMP and other molecules, other proteins, and lead to activation of those downstream proteins. So here you have the protein kinase A molecule It works as a tetramer. It has two regulatory subunits attached to two catalytic subunits. The regulatory subunits are inactive until cyclic AMP is bound to them. When cyclic AMP binds to the regulatory unit, it causes the catalytic units to become separated and activated on their own. And these regulatory units can no longer bind to them. The catalytic units can then go activate other proteins through their catalytic activity. Yeah. This way of activation leads to transcriptional change at the end of the day. The activated protein kinase will usually go inside the nucleus. It translocates into the nucleus where it can directly activate transcriptional regulators by phosphorylating them, right? That's what it does because it's catalytic. Activity is kinase activity. So it's going to go inside, it's going to activate these transcriptional regulator by phosphorylating them. Once you phosphorylate these transcriptional regulator, they are going to bind to their operator regions within the genome and activate those target gene transcription. So a lot of people talking and sending processes along. Proteins. That's people. Okay, so here are some specific cell responses that are mediated by cyclic AMP. Epinephrine in heart, skeletal, and fat muscles are all regulated by cyclic AMP. Each one is going to do a completely different function, but they are all working through this process. Similarly, adrenocorticotropic hormone, ACTH, in adrenal gland is also regulated by cyclic AMP production and activation, and it leads to cortisol secretion and the flight and fight response. So here is an example from that with the adrenaline in our body. So if epinephrine binds to our GPCR, right, this is again the G protein coupled receptor, it's going to bind to an activated adenyl cyclase in this case. That's that intermediate that's going to activate CAMP. And the alpha subunit is going to activate it, leading to ATP hydrolysis and making into cyclic AMP molecules. Cyclic AMP is then going to bind to protein kinase. Protein kinase will become activated and it's going to go down and it is going to phosphorylate other proteins, in this case, phosphorylase kinase. And eventually it's going to activate through, again, ATP hydrolysis, a glycogen phosphorylase, which does exactly what it says. It breaks down glycogen. And that will lead to the breakdown of glycogen as a byproduct of this epinephrine coming in. Yeah. You guys should see at this point the general pathway, the way it works, right? That you have a extracellular molecule binding to that G protein coupled receptor. It's activating the alpha subunit and the beta gamma subunit, and then they're binding to target proteins, and then activating some second messenger in large amount to amplify the signal, which then is going to lead to the target response. Yeah. So another cool thing is that some dissolved gases can also be important, be enough to activate intracellular enzymes through GPCR signaling. So you can see that in obviously our blood system and also in our musculature, usually in our endothelial cells. You have your smooth muscle cells on the outside, you have the endothelial cells, and then you have the blood vessel inside with the blood flowing through it. Acetylcholine binding to these receptors in our endothelial cells, so in this second layer, right, that is in contact with the blood, can lead to an increase in calcium levels. That calcium ion activation can help to release nitrous oxide from the endothelial cells. These nitrous oxide molecules will rapidly go across any plasma membrane that they see right next to it, in this case, a muscle fiber or muscle cells. So when they go through, they have the diffusion across the membrane, they will activate, again, a GTP molecule and turn it into a 6-GMP in this case, leading to relaxation of those smooth muscle cells. So if those muscle cells relax, what's going to happen to the blood vessel? It will dilate and allow more oxygen to go through. So this is one example where you can get these small levels of changes in the oxygen concentration or nitrogen oxide or carbon dioxide can lead to this rapid response so that it can relax and dilate the blood vessels and allow them to get more. So this, you think about it, this has so many steps involved, you would think that it would take a long time. But in reality, these GPCR triggered intracellular signaling cascades are super quick and they can follow, they can have almost instantaneous response happening from beginning to end. So it is a very sensitive and very fast in the way that it functions. A single molecule activation, so rhodopsin is another, by the way, GPCR. It's a G protein coupled receptor. When activated by light, it activates 500 G protein molecules, which each one of those is going to activate 500 cyclic GMP phosphodiesterase and then 10 to the 5 cyclic GMP molecules. So within a very quick response, it's going to, within a period of 1 second, you have activated a humongous amount of cells, many number of cells to signal that relay, send that signal to the brain, right? Okay, so the second part, we have enough time to go through receptor tyrosine kinase. So the second part of our thing is going to be looking at enzyme coupled receptors. So remember, there were how many classes of signaling in the membrane? 3. What were they? Remind me.
Protein.
G protein, ion coupled, and this enzyme coupled. And we kind of talked about a little bit already with the G protein coupling with an enzyme and G protein coupling with an ion channel. But now we're going to talk specifically about a receptor tyrosine kinase, which are enzyme coupled receptors. They are not involved in G protein at all. These are just receptors themselves that can activate other enzymes and then they can activate downstream signaling. So these RTK receptors, the receptor tyrosine kinases, they typically function as dimers. So 2 molecules have to come together to be activated. In a normal membrane, you will see inactive state, they will be separated, but close by, nearby to each other, but separated. When you have a signaling molecule that comes in, the signaling molecules for these usually also come in as dimers, where each one of them is going to bind the receptor kinase, tyrosine kinase, each molecule, and it will allow them to come together. When they get together in their activated form, it stimulates their kinase activity and causes several phosphorylations to happen all along their intracellular portion. Each one of these phosphorylations lead to a different mediator protein getting activated as a result. So a single receptor kinase, tyrosine kinase, when activated, can activate many different types of molecules as a result of it right at that top, because each individual phosphorylation lead to a different response. So here, for example, it shows you how your individual ones, there are 3 different ones shown, and each one of these 3 is going to have intracellular signaling proteins that is going to activate a different pathway, right? In many cases. So here is a 2 common receptor tyrosine kinases that we look at all the time. One of them is PDGF beta receptor, and the other one is the epidermal growth factor receptor. These several binding sites and some molecules attached to them. When these are phosphorylated, this leads to this particular mediator protein getting activated. Again, you don't need to know that what proteins are getting activated, but the fact that individual phosphorylations lead to activation of different mediator proteins. And so if there are 5 different phosphorylations, you may be getting 5 or more activated proteins as a result from it. And you can see, like a couple of them, both of these phosphorylations lead to PI3K gamma activation. This particular phosphorylation leads to GAP protein. Remember GAP, G protein coupled receptor. So this would probably then link with a G protein coupled receptor. In that case, others are independent of G protein. Here again, with the EGF receptor, individual phosphorylations are leading to activation of different activator proteins, different mediator proteins. So here is an example of one thing that it can do. And we just kind of showed it in the last slide. Receptor tyrosine kinase can activate G protein coupled receptors, right? That's one thing that it can do. So they can be used to activate that. So here is an example of one thing like that. A common protein that is a G protein that directly interacts with tyrosine kinase is RAS protein. And RAS is, by the way, also involved in tumorigenesis. So that is something important for that as well. The RAS protein in its inactive form is bound with a GDP. So it's like that alpha subunit of the G protein complex. The signaling molecules that you have over here, one of these signaling molecules, when activated on when the RTK is activated, one of those signaling molecules is an adapter protein that interacts with a RAS-GEF, the guanine exchange factor. What's it gonna do? Well, it's gonna activate this RAS protein by exchanging the GDP to a GTP when activated. When it does that, now you have the activated RAS protein, and it's going to send signal downstream through activation of other mediator proteins. RAS protein is involved in cell cycle regulation, so that's typically what it then converts into, is a signal towards that. So here you have that. RAS protein, the way it works is by activating another set of pathways. Once activated, it will activate MAP kinase activity. And you see how it is like, it ran out of names, I guess. So it's activated MAP kinase, and then that will activate MAP kinase, and that will activate MAP kinase. And that will lead to transcriptional regulation. So there are two activated MAP kinase pathway lead to two main effects, changes in protein activity and changes in gene expression. The changes in gene expression are regulated through two different transcriptional regulators that can lead to regulation of cell cycle at the end of the day, okay? Another similar type of a pathway that receptor tyrosine kinase activate is once they're phosphorylated, they can directly interact with the inositol phospholipids and release that IP3 molecule from them, that phosphorylated inositol phospholipid, through activated PI3 kinase. So this is activating the, into an IP3 molecule that can obviously act as a signaling molecule in itself. And that IP3 molecule interacts with activated protein kinase 1 or activates protein kinase 1, which activates AKT, which activates further survival mechanism. AKT is responsible for survival of cells, or is very important in survival mechanisms, a lot about cells. So two main things that it is controlling through RTK activation is RAS protein, which is going to help control cell cycle, and AKT, which is going to help control survival. Okay. So RAS with cell cycle and AKT with survival. And this will become important also in your lab experiments that you are doing. 2020. Okay. So what does AKT do? Well, it promotes cell survival. How does it do it? It does it by interacting with other proteins inside the cell intracellularly. Activated AKT leads to phosphorylation of bad protein. When phosphorylated, bad protein cannot bind to BCL2. So the BCL2 protein, when bound to bad, is inactive and cannot work. When bad is phosphorylated, it disrupts that binding. Now BCL2 is free to inhibit apoptosis, and it inhibits apoptosis by binding to this another protein called cytochrome C and keeping it in the mitochondria. And we kind of already talked about it a little bit in lab, I know, with the apoptosis pathway. Remember that apoptosome that was forming in the intrinsic cell death response, you had the cytochrome C releasing from the mitochondria. Well, this is how it was going about in the reverse. Okay. AKT can also have, again, a response on the cell cycle as well as a secondary target. And in those, the receptor tyrosine kinase, when activated, will activate PI3 kinase, which will activate AKT. And then several things downstream, several mediator proteins downstream, it will activate this other protein called TOR. The TOR protein can stimulate protein synthesis and inhibit protein degradation. So it's not directly affecting cell cycle, but it's kind of, again, going along with the survival theme, that it's not letting them die and giving them that extra time so that it can have a longer survival and eventually maybe be able to grow and survive and re-enter cell cycle. Now, TOR is a target of a common chemo drug called rapamycin. It's a targeted therapy. Rapamycin essentially works by inactivating TOR. So if you inactivate TOR, the cell cannot go into the survival mechanism, right? So it won't be able to inhibit the apoptosis like we did before. So at the end of the day, what you should get out of today's lecture is that while we have all these signaling pathways individually present, they are also interconnected. There's no signaling pathway that happens in isolation. Everything is crosstalk between each other, whether it is signals coming in from outside or signals within the cell. All of them are constantly having interaction with each other to lead to the final response that the cell undergoes, whether it be growth, replication, survival, or even dying. All of those are regulated by the crosstalk between these various pathways that you are going to see. And as we talked about, the two main ones with the GPCR or the RTK systems. Okay. So, there's not a lot of things, yeah. Well, sometimes these can take just weird. So one of those things, one of the way that we talk with each other is, and this is the pathway that we did not talk about yet as much in detail, was remember that contact dependent pathways, and we talked about the notch delta signaling pathway. So in that case, you don't have all these GPCRs and RTK quite functioning the same way. The notch receptor itself, in this case, in these contact dependent signaling, can act as a signaling molecule in itself. It is the one that's going to regulate growth or activation of the gene expression. So the ligand delta, when it binds to notch, activates it, leads to cleavage of its cytoplasmic tail. The cytoplasmic tail, when cleaved, translocates into the nucleus and directly binds to notch response elements within the genome and transcribe genes that are regulated by that. So this little portion of the notch tail, that's what acts as a transcriptional regulator in this case. Okay, and then obviously, so as we talked about in the beginning, you don't have to have the intracellular signaling proteins just responding to extracellular signals. In one way, they can all have crosstalk between each other. So you have various receptors. Each one of them is going to activate one or more pathways. Some are going to be activating them. Some are going to be inhibiting the same pathways. So at any given time, the final response that the cell shows is based on the amount of activation versus the inactivation, right? The inhibition versus the activation, and whichever side gets to be the higher one, that's the one that shows up within the system. Okay, all done for today. We actually got through a lecture and then a class this time, which is fun.